Hello there. Before we jump into the specifics of clinical research, I just want to take a minute to let you know that this video is part of a more comprehensive class that describes pharmaceutical R&D from drug discovery right through to marketing. If you'd like to know more about that, click on the link below to check it out. So let's just put the clinical studies in context in terms of the overall development timeline for a new drug. Well, once a candidate is chosen for development, an IND is submitted to the FDA, and unless the FDA issues a clinical hold, 30 days after the IND is submitted, the clinical studies can begin. And the clinical studies are done in 3 phases. Traditionally, phase 1, phase 2, phase 3. And we're going to talk about each one of those phases individually now. Let's look at the individual phases of clinical studies. I'm sure you will come across these terms, but there are 3 phases that are distinct, and each has its own importance and really is indispensable. First is phase 1 clinical trials, and these are really intended to evaluate safety tolerability. So what's done is the drug is dosed in different groups of people, single dose at higher and higher amounts. Basically, it sounds funny to say until there's somebody says ouch, until there's some kind of a signal where, oh boy, now we're running into trouble. And people are very carefully monitored. It might be, for example, let's say irregularity in heartbeat. That's a simple thing to evaluate. And so, or it might be a headache. And when we're seeing these signals, then the doses basically say, okay, that's as high a dose you can start with. And remember, the initial dose is based on a no adverse effect level that is found in the animal studies. That's the lowest dose that we start out with in the human studies, phase 1. Okay, but here we're looking at, it's very important to remember, we're looking at healthy volunteers. I have it underlined here because this is something that kind of doesn't make sense to a lot of people, but healthy volunteers, not people who have the disease that we're trying to treat, because we're just trying to see if the drug has any, I should say, characterize what the adverse effects are in humans. Okay, and I do want to mention here that traditionally men are used in phase 1 studies, and also women of non-childbearing potential. In other words, young women, for example, have had a hysterectomy. And the reason for that is not so much that, people who are involved in this business are sexist, because many of the people doing the research are women themselves, but because men produce sperm on a continual basis. So if you give a drug that damages the sperm, well, a few weeks later, that sperm is gone and there's new sperm being formed. But eggs, a woman's eggs, are there through life. So if you damage them in some way through a study drug, that's going to be something that's going to be with that woman for the rest of her life. So this can be a contentious issue, but that's basically the idea. Now, typically, phase 1 studies are not placebo-controlled or randomized. What I mean is that we don't have a cohort of patients that are dosed with a sugar pill or a non-active pill. And patients, as they come into the study, they're not randomized. That randomized means that you select people to be put in one group or another. Since we only have one group, that is the people taking the drug. There's traditionally no randomization. We'll talk about that in more detail later. And a starting dose is typically a fraction of the highest no effect dose to NOEL that we talked about in animals. And phase 1 studies can also include the radiolabeled ADME study that we spoke of in animals. So a study with radiolabeled carbon-14 is typically done in humans early on, just to characterize the pharmacokinetics, which I should have mentioned in the first point here. Also, the pharmacokinetics are very important in phase 1 study. Does the drug have like a a half-life of maybe 15 minutes or an hour. If you're trying to develop a drug to be given once a day, that would essentially preclude its further development. It would have to have a half-life somewhere between 4 and 8 hours for once-a-day dosing, for example. Or if it the pharmacokinetics, if it's completely eliminated by excretion in urine, you would know that, oh, we have to be, later on down the line, we have to make sure we evaluate people who have kidney problems, renal dysfunction. So again, just to review this, safety, tolerability, pharmacokinetics are what's involved in phase 1 studies in normal volunteers. Phase 2 studies then are focused on evaluating efficacy, getting a preliminary idea. Does the drug do what we want it to do in the target population of people who have the disease state that is intended to be treated? Here, there's typically just maybe 100 people, not too many more. Sorry, I should have mentioned in phase 1, there's generally less than 20 people involved in that kind of study. In phase 2 studies, we're giving higher and higher doses now to look at what dose will give us a beneficial effect without too much toxicity. Okay. And again, this is done in patients. And one of the main things here is to evaluate what is a reasonable starting dose to use in phase 3 studies, and what you're going to hear about in a minute. But phase 3 studies are huge studies, and you want to make sure you go in with the right dose. That is a very, very important thing. So phase 2, are we seeing any signals that the drug is working? If in a phase 2 study you look at 100 patients and none of them seem to have the effect, let's say it's a drug for diabetes and nobody has a lowering of blood glucose levels, typically the investigators would just stop the study. The development team would say, let's terminate the study right there. But if things look good, then we continue to phase 3 studies. Now, phase 2 studies, each one of these terms is very important to understand. Let me take a minute to explain it. Phase 2 studies and phase 3 studies are typically run double-blind, randomized, placebo-controlled. Double-blind means neither the investigator or the patient getting the drug knows whether the person is getting the placebo or the active drug. Okay, that's very important. Okay, that's double-blind. Randomized means that as the people are recruited for the study, which one go to the placebo and which one goes to the active drug is a random process. There's no one saying, let's put this one in this group and this one in this group, which might bias the results. Okay, placebo-controlled obviously means that there's some of the patients who are getting a placebo, and it's fascinating the way this works out, because sometimes it's very difficult to really package the product so that the placebo and the active look exactly the same. It would have to be, for example, both of them in a gray capsule to make sure people don't know which one is which. Active control only means that in the case where there is a drug that's known to be effective for the indication, it's really not considered ethical to give a group of patients a placebo. So the control arm would get what's the sort of the state-of-the-art drug, and then the active arm or the experimental arm group of patients would get the new drug, the drug being developed. And then we have our phase 3 clinical studies, which are really essentially larger versions of phase 2 at a given dose, though. Okay, so phase 3 studies, involve thousands of patients, multiple sites. What we mean by that is that there might be 100 patients at the Cleveland Clinic and 100 patients at Johns Hopkins. These are typically done at large sites that do research studies, because you have to be able, for if nothing else, recruit patients who have a particular disease state. And the results of this are really what goes to the FDA and allows them to make a decision whether or not we're going to go and approve the drug. Okay, it really allows the sponsoring company to do a risk benefits analysis. If this is a drug for cancer, obviously you can accept much higher toxicity, much greater risks in order to get an extension of life, for example. Whereas if it's something for obesity, it has to be really squeaky clean and have minimal risks in order to have that benefit of reducing weight. Okay, so that can be assessed only by the phase 3 trials. In principle, if you could just do phase 3 trials, you wouldn't need to do anything else. Everything else is just leading up to the carrying out of the phase 3 studies. These are what are called pivotal studies, by which the decision is made by the FDA in order whether or not to approve the drug. And typically these are, again, randomized, placebo-controlled. Yes, that's kind of considered the ultimate way to run a clinical study, because it eliminates or at least minimizes any chance for bias. And typically there are two separate studies with thousands of patients that are done in order to register or in order to file an NDA, a new drug application, at the end of the day. Now, one additional type of clinical trial that goes on or is involved in the drug R&D process is called a phase 4 clinical study. And these kind of studies are basically post-marketing. These are done after a drug is already approved and on the market. Okay, and so they're distinct from phase 1, 2, and 3. Okay, phase 1, 2, and 3 are pre-marketing studies. The objective here is to get some idea of a particular risk, for example, that may have been seen in the pre-marketing clinical studies. Occasionally, the FDA will say, we're a little concerned about peripheral neuropathy that we saw in a couple patients in your earlier studies, maybe patients who are African American, for example. And so now they'll ask for a study to be done in African Americans to look for peripheral neuropathy. Okay, and that can be done even though the drug is approved. This is a post-marketing phase 4 study. This may involve thousands of patients, may be done years after the filing. And another very important use of phase 4 studies, probably the most important, is its use for other indications, extending the indications, extending the utility of the drug. For example, when studies are being done to show that a drug, let's say, is useful for diabetes, for reducing blood glucose. And one of the effects that's noted is that, oh, how do you like that? It turns out that the drug also causes people to lose weight. Well, that was not one of the primary endpoints being evaluated, so it may be not that clear what the effect on weight is, but it seems like it has an effect on weight. So the sponsor might want to go back now in phase 4 studies and go back and look specifically at the effect of that drug on weight loss in a controlled setting. Okay, this would be compared to placebo, for example. And here's the important thing to remember: until the drug is approved for that additional indication, the sponsor cannot even suggest to a provider, a physician, that the drug is useful for that indication. It's a little bit tricky how that actually turns out in practice, but that's the idea of a phase 4 clinical study. This table just puts together in a concise form pretty much everything we've been talking about up to now. You can go back and refer to it if it helps you. And here are some couple of references that might help if you want to look into some of what we've been discussing in some more detail.